Good evening. My name is Steve Martin. I'm a faculty member in Material Science and Engineering. And tonight, it's my great pleasure to welcome you to our second Sigma Xi lecture for fall of the 2010 academic year. And we will have another two talks next spring. Each fall and spring semesters, we have two lectures. We have a national honors lecture, and we have a local honors lecture. And so it's a great privilege for us as a Sigma Xi to offer these seminars and to engage the greater research community at Iowa State. Sigma Xi is a national and international organization focused on research, and it cuts across all disciplines of research, everything from, of course, engineering to biology, but also information and history. So it's a very, very broad organization and a very wonderful organization to be a part of. Not the least of which is you get to come to great lectures like tonight. So keep aware of the advertisements. Our next seminar will be in February, and the announcements will be out. We'll be also here in the Memorial Union, typically on a Thursday night, typically at about 8 o'clock. So if you're interested in more information about Sigma Xi, I suppose there's a few members of Sigma Xi in the audience. If you're interested in Sigma Xi, it's open to, of course, researchers of all ages, students, graduate students, faculty members, and so forth. So please look at Sigma Xi. It's right on the ISU homepage, and you can look into our activities and becoming a member of Sigma Xi. Of course, for students, there's very low reduced rates for membership. It's a great pleasure for me to introduce, I'm the president of Sigma Xi. Diane Burt is vice president of our organization, the ISU chapter of Sigma Xi. Diane, Professor Burt, is a distinguished professor in the Department of Food Sciences and Human Nutrition. She's also the director for the Center for Research on Botanical Dietary Supplements. And so she also has a great level of research experience in food and food supplements and dietary health. And so with that, Diane then will introduce our speaker. So Diane, please.
Thank you very much, Steve. It's a great honor for me to introduce Dr. Vasant Hanavar. He has a Bachelor of Engineering in Electronics Engineering from Bangalore, India. He has a Master's in Electrical and Computer Engineering from Drexel and a Master's and PhD in Computer Science from the University of Wisconsin-Madison. Here at Iowa State, he is a professor of computer science. He founded in 1990 at Iowa State the Artificial Intelligence Research Laboratory, and he currently directs that. And he also directs the Center for Computational Intelligence, Learning, and Discovery, which was founded in 2004. He has a very long list of research interests. I think that we'll probably not go through the whole list. Obviously, artificial intelligence, machine learning, knowledge representation, bioinformatics, computational biology, and the list goes on. He's published over 200 research articles in refereed journals, conferences, and books, co-edited 6 books. With that, I will leave it to Professor Hanavar to speak to us about algorithmic thinking in biology.
Okay. So most of my work is done with graduate students, so here's some of the people that I work with. So today's talk, I want to convince you that computer science is not just about these computers. Computer science has an impact on other disciplines, and today's focus is on biology, but the same case could be made for other disciplines. So the first part of the talk, I'll try to convince you that computation is important, and it's important for all sciences, and in particular for biology. And then I'll give a few examples of sort of the consequences of this way of looking at other sciences, in particular biology. Okay, so let's start with just something to get us warmed up. So this is something that appeared in a journal, a biology journal, a few years ago. So can a biologist fix a radio? So how would you go about fixing a radio? Well, here's a possible way to go about fixing the radio. So you open up the radio, find all the different things in it, then you classify the things that you see, then maybe even try changing, replacing some component, see what happens. If it's still playing, then you assume that that wasn't probably, the color of that component was not probably critical. And I wouldn't dare put this up if it wasn't for the fact that this was actually written by a biologist. Okay? Otherwise, it would seem a little odd that a computer scientist would put this up. Okay, so, and if we, this is a quote from Rutherford, who said, all science is either stamp collecting or physics. And biology, one might say, and this is, of course, a caricature, so I'm going to exaggerate a little bit so I don't get offended. Biology is like a bit like physics before Newton. And physics before Newton was a descriptive science. Right? So you could throw something up in the air, you could write down in your diary, I threw something up in the air, it came down, but you had no way of predicting what the trajectory was going to be. So there was no really, the science of physics didn't exist. And Newton co-invented calculus, which gave for us, for the first time, a way to talk about things like rate of change, and gave us the language which allowed us to develop, actually, Newton to develop classical physics, which took about 200 years to fully that particular thing to happen. So biology is at a stage where physics was right about the time Newton invented calculus. Because biology has been a descriptive science, so you collect, catalog, and describe things that you see, biological objects, biological phenomena. And this goes from the early days when taxonomists went and cataloged species and their relationships, to more recent times where we sequence the genome and describe what we see. But unlike physics, in biology there hasn't been quite yet any sort of predictive. It's not a predictive science, so I cannot tell you what will happen if I manipulate certain genes or how it will affect the organism. I can't. So we don't have anything like physics for biology. So it's been a descriptive science, and advances in biology have been limited in part by our instruments of observation, and this is critical for any sciences. You have to be able to observe before you can do anything more. So you first observe, then describe some things, and then try to build predictive models. And then there have been limitations on all of these things. But things have been changing in the last several decades. So biology used to be what people call a data-poor science. So most of the time people studied a single gene, maybe for their entire careers. But now we have at least the instrumentation that allows us to gather lots of data about specific biological systems. So if you want to transform the biology then from stamp collecting to physics, from a descriptive science into a predictive science, then we need methods for constructing models from data, inferring consequences of some hypothesis that you have on this large scale data, generating hypotheses that you can test against data that you have, and then designing optimal experiments, and so on. And I would argue that over here, computation plays a central role. So, since this is a diverse audience, before I go on, first let me say a little bit about computation. So, this computation, everybody knows about computers. We all use computers. But computation, the discipline of computer science, has a very rich intellectual history. We started out with Hilbert presenting this decision problem, which was a very simple. Hilbert is a mathematician who is more famous for the problems that he posed than for the problems he actually solved. And many of his problems are still being solved by mathematicians. But one of the problems that he posed was, is there an effective procedure for deciding whether some conclusion logically follows from a set of assumptions? So given some facts, is there some procedure that tells me whether some conclusion follows from the assumptions? It's a very simple question. And Alan Turing tried to address this question. And before he could address it, he had to formalize what was meant by an effective procedure. So what is an effective procedure? Asking your grandmother, staring at the sky. What is an effective procedure? So, and to make the long story short, Turing invented a little gadget called the Turing machine, which had a handful of instructions, which had a tape on which you could read and write symbols, and it followed a simple set of instructions to move the writing hand left to right and read symbols out of it. And then basically he suggested that computation, or this effective procedure, is, for all practical purposes, what this Turing machine does. It's an effective procedure is a program for this Turing machine. And what it does is essentially transform a string of letters into another string of letters according to some rules. And that's all there is. Computation is essentially that. And many other people tried to come up with alternative formalizations of this, and they were proven to be equivalent. Okay, so there's something fundamental. Computation is a fundamental entity. So, so this led to the Turing thesis, which, in sort of layman's terms, is anything that you can describe can be described by a program for this Turing machine. And Turing machine is essentially what you have on your desktops. They're just faster versions of Turing machines. And in fact, they're more limited because they have limited memory. Turing machine assumes unbounded memory. Okay, so if you take this seriously, then theories and models in any discipline take the form of algorithms. And artificial intelligence was one of the fields which actually took this pretty seriously, and it works under this assumption that computation for cognition or thinking is what calculus gave for physics. So they try to describe models of thinking in terms of algorithms, programs. And computational biology also takes this sort of thing seriously. So by analogy, I can say computation is to biology what calculus is to physics. So in other words, calculus gave the formal language for describing physical phenomena. So here, you can think of algorithms or programs as giving the language for describing phenomena in biology. So that's sort of the main idea. So computational biology, then, is a marriage of biology and computing. And it rests on this premise that computation provides for biology what calculus provided for physics. It's an approximate analogy. And as a means of transforming biology from stamp collecting to physics, descriptive science, to interpretive science. Yeah. So this means, an implication of this is that biology now becomes fundamentally an information science, because you're going to be the basic objects here will be theories about information processing. So for example, if you look at the genome, so our organisms in general you can think of them at a certain level of abstraction as mutable, self-reproducing information systems. So you could look at the genome, you could think of it as a program which encodes the structure of the organism and to some extent its behavior, which is going to be influenced by interaction. So, and they acquire information, so through learning, adaptation, and evolution, they transmit information. So this is at a certain level of abstraction. So this doesn't mean that physics and chemistry and everything else is not important, but when it comes to describing phenomena at the level of information, then you can use computation as a framework to describe. So biology is fundamentally an information science, and that in this point of view, and then the grand challenge of biology is given code that's written in some unknown programming language, running on some unknown computer architecture. Our challenge is to determine the syntax and the semantics of the language. In other words, what does this program mean? What does it do? Right? And its editing history. In other words, how did it come to be? Right? Because it's through a process of evolution. And so on. Just to give a different example, suppose Martians came down to Earth and they could look at Shakespeare's work, but they didn't know anything about human language. And they had to figure out what English language, how English language operated. So they had to figure out English language. And think about how challenging it would be. So that's really the challenge of biology, because we have a language, we know the alphabet, but we don't really know the rules of grammar or what things mean, what they do, what genes do. So figuring that out is like figuring out the syntax and semantics of some unknown language from essentially by observing, some things written in that language. So just some terminology. So when we talk about the genome, it's basically we refer to the entire, so the genetic code sequence, which is written, so strings in a four-letter alphabet is ACGT. In the case of viruses, it happens to be a slightly different alphabet, the RNA alphabet. And just some very basic biology, since this is a diverse audience, each cell has identical DNA. So your skin cell and your cell in your heart and your kidney, everything has exactly the same DNA. But still the cells are different. So what makes them different? Well, this program, if you think of it as program, then the program, what the program does depends on the input sequences. So if you had a computer program, it takes a branch based on some condition, and that condition might be met if you have some particular input. So this cellular differentiation is essentially a response to the signals that orchestrate this developmental program. So trying to understand that, the response of this program to different conditions is a huge problem, so the program of life. Transcriptome refers to the full complement of genes. So genes get transcribed into messenger RNA, which then get translated into proteins. And the proteome is the full complement of proteins that is produced. So most of the work in your body, hard work in your body, is done by proteins. And then there's this term interactome, which refers to the full complement of molecular interactions. Proteins have friends, they interact with other proteins, and that's how they function. So these interactions can be modeled by a network. So for example, a graph, that the nodes have proteins and interactions. And the techniques that we use there are no different from what you would use to analyze a social network or some other type of network. So one of the challenges is sort of relating these different levels. So for example, the program at this level, how does it get translated into this, and linking up these different levels of abstraction. So if you take this approach seriously, then we will have a theory. So protein folding is what we refer to. So there is this linear sequence of amino acids which is obtained from the genes, and it folds up and forms a three-dimensional structure for protein molecule. So and it goes through this process called protein folding. So one of the questions is, how does this linear sequence know what three-dimensional shape to take? So presumably the information is encoded in this sequence. So we will have a theory of protein folding when we have an algorithm, a program, that takes a linear sequence of amino acids and generates from it a description of the 3D structure. So if you can do that, if you can develop such a program, then you understand how protein folding works at that level of abstraction. So, and the same sort of story can be told about other problems in biology. So, and you can take a similar sort of line of reasoning, and this has affected other disciplines. So I'm going to skip this because the focus here is on biology. So, so this allows us. So computational models or programs are cartoons, right? So they're not necessarily reality, but they describe certain aspects of reality, relevant aspects of reality. F equals ma is a cartoon. It's coming about physics. Now, if you're interested in phenomena at really submicroscopic levels, F equals ma is not going to work. It works at this, sort of classical physics level. So any model that we develop is not so it's not reasonable to ask if a model is true. This is not about what's true. It's about whether this model actually helps explain some phenomena at the right level of abstraction. So in the case of biology, you can come up with models or cartoons that describe basic entities like DNA, RNA, genes, proteins, and so on, the properties of these, relationships between those, so interactions. And depending on exactly what we are interested in, you can come up with different types of models. These models take the form of essentially programs. So, so this is sort of the big picture, which connects computer science with biology. And one could argue that computational models are what allow you to transform biology from stem collecting to physics, in broad sense. So to sort of wrap up this section of the talk, the main idea is we understand a phenomenon when we can write a program that models it at the right level of detail. So I understand protein folding when I can come up with an algorithm that takes a linear sequence of amino acids and generates from it a description of the structure. So, and when theories take the form of algorithms, and the field that studies algorithms happens to be computer science becomes relevant to the sciences. And this has led in recent years to the birth of many different disciplines, and they all have computational added to whatever it is: computational biology, computational chemistry, computational economics, computational social science. Okay. And this also has implications for both research and education. So, in a sense, if you take this seriously, any literate person has to know something about computing. Not merely the use of computers, but the ideas, the concepts of computer science. Okay, so now let me give you some examples of this sort of thinking at work. So starting with something that happened about the same time as computer science was born, 1943. And this was partly had to do with artificial intelligence, partly with biology, because this started with trying to understand how brains work. And and brains are made of neurons. And so McCulloch, who was a neurophysiologist, and Pitts, who was a mathematician, who heard about computing, thought they could take a computational sort of view of what goes on in brains by trying to model neurons and brains. Okay, so, and there's a very famous seminal paper, Logical Calculus of Neuronal Activity, and so, here's the essentially what they did. So there was a little bit known about how neurons work in 1943. A lot more is known now, but at the time. So this is probably, if you took a biology course in high school, you've heard that neurons have a cell body. They receive input from other neurons to the so-called synapses, and that changes the electrical potential of the cell, and that causes a stream of pulses to go down this axon. It turns out, by the way, that this physics of this is exactly the same as physics of coaxial cables. There is this fact that insulates it. So this is obviously a very complicated system. So you can describe it using differential equations and so on. But you can also describe it using a very simple sort of cartoon, which leaves out a lot of details. So one such cartoon is this one. So where you can model the different inputs to this neuron using some variables, x1 to xn. The interactions through synapses, these contacts here, by some weights, some numbers. And the fact that this adds up these signals and eventually sends out pulses by, instead of doing an integration, which you would do if you modeled this using continuous types of mathematics, here instead you would have a summation, and then if the sum of the weighted sum of the inputs exceeds some value, which we call the threshold, then the output is high. So these are pulses that go out. So you can think of it as a switch. It adds up the inputs, and then if the inputs exceed a certain value, it sends an output. Very simple gadget. So the output is a 1 if the weighted sum of the inputs is greater than 0. It's 0 or minus 1 otherwise. So it's a two-state device. Okay, so if this cartoon was a reasonable model of neurons at some level of abstraction, you can then ask, well, what is this neuron capable of doing? And for one thing, it has some interesting connection with geometry. So if you have this sort of linear summation, so if it's greater than 0, something happens. If it's otherwise, something else happens. So what if it is equal to 0? So by setting this weighted sum equal to 0, I get essentially an equation for a line or a plane in some high-dimensional space. So, and for any input that falls on one side of the plane, this device produces an output of 1. And for inputs that fall on the other side of the plane, this device produces an output of 0. Okay, so if you think of these points in the space as modeling inputs, so it could be a signal that you receive from your visual system, this can be used as a pattern classifier. Right? It classifies its inputs into two categories. One when it says output is a 1, one when it's produces an output of -1. So it can be used to classify things. Now, the precise classification depends on where this line or plane is located. Right? If I change this, then the classification changes. And the position of that plane and the parameters that define it are essentially the weights of it. Okay. So there's some interesting connection with geometry and also pattern classification. So we can use this as a classifier. It also has some interesting connection with logic. Okay. Again, a simple cartoon that was based on this abstraction. Well, by choosing the weights appropriately over here, I can get it to compute certain logical functions. So, for example, if I set the weights in a certain way, and I do this computation. If you look at this output, this is essentially the logical OR function. If you think of -1 as logical 0 and +1 as logical 1. So you can get it to compute some simple logical functions. And it turns out that by choosing the weights appropriately, you can get it to compute ANDs, ORs, and NOTs. If you know if you have had any exposure to Boolean logic, one of the very basic results is that if you can compute ANDs, ORs, and NOTs, you can compute any Boolean function by combining them. So there's a trivial theorem that one can prove that says that for any Boolean function f, there is a network of these little neurons that would be able to compute any arbitrary Boolean function. So once you can compute a Boolean function, it's a very small step to building finite state machines. These are machines that can go through a bunch of finite number of states and receive inputs and respond to them. Okay. And that's exactly what we have in these boxes here, computers. So all we need is to introduce some delay units, and you can do that. And if you give them access, if you have such a network, which is a finite state automaton, and then if you give it some scratch space to write and read from, then you get Turing machines, general purpose computers. So this humble neuron, which is based on this cartoon of how neurons actually operate, can be used to build general purpose computers. So now all of this came from, all of these results sort of follow from just taking this neuron, abstracting away a lot of the details of how neurons actually function, coming up with a very simple model, and then asking questions about what is this model capable of. So now, network of neurons can compute arbitrary Boolean functions. Now the next question you can ask is, well, if it's neuron, can it also learn? In other words, can it change the function it computes? Or can it change the way it responds to its inputs? And the answer turns out to be yes. One can come up with a very simple learning algorithm that basically modifies the weights based on the inputs and the class table that you have. And it turns out that after a finite number of iterations, if it converges, it will converge to the right boundary that would get the inputs correctly classified. So anytime you have a set of data samples, in this case, for example, I've labeled these, and if I had some data samples taken from this space, and I give it to this algorithm, then it can adjust this decision boundary so that it can correctly classify data samples. So that's a simple form of learning. Now, this was all 50 years ago, and there are much more sophisticated learning algorithms now that can work with more complicated sort of learning problems. But the basic idea is the same, which is have a data set of labeled samples, and you can get this machine to learn. Okay, so that's one example of, and this is if the situation gets more complicated, you come up with, you have to go through some additional steps. And the last 30 years, this has really led to a theory of learning machines and the far more sophisticated algorithms that can learn from data. So that's one example of a computational or algorithmic thinking in biology in the very simple context of looking at how neurons function. And that leads to, and that led to the development of an entire new field, computational neuroscience, where you have computational models of neurons and brains and neural circuits. And it also led to the development of an entire field of subfield of artificial intelligence, learning systems, learning lineage. Started out with this one simple exercise of taking a neuron and trying to model it using some simple computational ideas. Okay. Now let me turn to some examples of if you have these learning machines, where could you apply them? Well, you can apply to a lot of different scenarios in biology. And so here's some examples. So one of the basic problems is when you have these proteins, they perform their function by binding to other proteins. Molecules function by interacting with other molecules. So one of the basic problems is, can you predict where the interaction sites are? Okay, so what makes a protein interact with some other protein? Well, these are essentially lock and key like mechanisms with some shape complementarity, and when they bind, so for example, if, the way the drugs function is by essentially blocking certain active sites. So if you could understand how these interactions work, then you can design better drugs, and you can do many other things. So one of the questions is, how can we predict interfaces from sequence or structure? So this can be approached using many different techniques, but one of the ways in which you could approach is by using machine learning. So you have a data set of well-characterized protein complexes, proteins that bind to other proteins, proteins that bind to RNA, proteins that bind to DNA. And from this, you can try to learn the general rules or some sort of a general predictive rules that would allow you to predict these interfaces. Well, so this is an example of such a study. So in this case, it was protein RNA interfaces. So you can go to the protein data bank, and from that you can extract a bunch of protein RNA complexes. And from that, because these are complexes, you know exactly where the interaction sites are. So you have a data set of protein RNA interfaces. So from this, the task is to learn what makes, to predict interface residues of some new proteins. Okay? So that's the problem. So here's again a statement of that problem. So you're given a protein. You don't necessarily have the structure of the complex. You want to predict which amino acids. So this is a 3D structure, but what you get is a sequence. Predict which amino acids participate in some protein interactions. So the guiding hypothesis is that the principal determinants of protein binding sites are somehow reflected in the local sequence. That the signal is there in the sequence. Okay. So if you're a machine learning person, you would approach it by generating data sets of known complexes. And then you build, so you use one of these machine learning algorithms, maybe something that's more sophisticated than the simple one that I told you about. And then you build a classifier, you test it on some new data. When you are confident that the classifier works, you can use it to classify interfaces. So now, there are many questions that arise in this setting. So for example, how do you take, what is the input to the classifier? So one simple way to deal with it is by taking the sequence of this protein, and for each one of these amino acids, you look at some neighboring region, and that would be the input. Or you could take the structure if it's available and look at some structural neighborhood and try to predict it, and so on. So suppose you do this. Again, skipping all the details of the algorithm and the primitives and so on. Just give you the result. So suppose you do this. You can do reasonably well. So you can get interface residues right with reasonable accuracy. This is not perfect by any means. But on the other hand, you have to keep in mind that we are using very limited information, essentially some neighborhood of the position on which you are making the prediction. And even if you don't quite get all the positions right, if you know approximately where the interface is, that's already pretty good for many of the targeting experiments. Okay? Or so you know roughly where the interface is, so you can try to design a drug molecule that binds to that region. Okay. So then you might ask, well, okay, so you can predict this. So what? What do you do with this information? So here's an example of what you can do with this information. So if you build these predictors, can it really give you some biological insights that tell you something about biology? Right? Or is it just an exercise in playing with data and building some models? Okay, well, here's actually a study where so this was an attempt to predict the binding sites between essentially a viral RNA, which is the horse version of the HIV virus, and the proteins that it binds to. And so there are predictions, and then there are experimental. So when you make some predictions, you can then target mutations at those predicted binding sites and see whether the binding activity is still preserved. And even though the predictions are not 100% accurate, you can get the general vicinity quite reasonably well, so that and the predictions are confirmed by the experiments, which means that these kinds of techniques can be used then to first of all focus the experiments, because experimentation, actual experiments are costly, so you can't mutate every possible site. So the predictions can be used to guide what sites to try to mutate. And if in fact the predictions are borne out by the experiments, then you can use that to take the next step, which is maybe design a drug that blocks that site. Okay, so these sorts of methods are quite useful. So, and in this case, the actual experimental work was done by Carpenter's lab here in the Veterinary Medicine College, and the predictions were borne out by experiments, and so that's a good thing. So now, one of the other problems is often if you're trying to understand protein-protein interactions, you take the molecule and then you do what's called docking. So essentially, there's a shape that this protein has. You have another protein, and you sort of move it around on this 3D surface to figure out where the energetically favorable conformation is, so that whether these two proteins can bind to each other. And this is a very hard computational problem, because you'll have to try every possible conformation if you're doing it blindly. So you have some 3D shape, you have some other 3D shape, and you're trying to figure out where they are complementary. So, or in other words, whether some particular lock would fit a particular key. So you'll have to try many conformations. Right? So here again, if you can predict interfaces, you can you can try to reduce the amount of computation that you have to do, and also improve the quality of docking. So here, so one of the things that you can do is if you predict the interfaces, then these docking programs generate some conformations. You can use the predicted interfaces, their agreement with the dock conformations, to rank the conformations based on whether the interfaces that exist in this conformations generated by the docking program actually agree with the predicted interface. If they agree, then it gets a high score. Okay, and it turns out that simply predicting the interfaces can improve the quality of results that are obtained from the docking program. So if you just took the ranking that's provided by the docking program based on some energy considerations, and you ask whether the ranking corresponds to the native conformation of this complex, there's almost no correlation. And this is because of many reasons. So first of all, we don't have the accurate energy function, so we don't quite understand all the physics there. Okay. So this and this is what you would see. So the dock conformation may not necessarily be whatever this docking program generates may not necessarily be the native conformation of the protein. But on the other hand, if you use these predicted interfaces, the correlation between the top ranking conformation, which is consistent with the predicted interfaces, also happens to be the native conformation. And this correlation is very strong. and we've tried, and this is very recent results, and we've tried many other ways of determining whether this is the ranking. So, for example, you can look at the RMS deviation from the actual structures and many other criteria. And it turns out that this simple technique of using predicted interfaces to rank the conformations improves the quality of the output that's produced by docking. And docking is a basic task that's used in drug discoveries. So again, computation plays a critical role in sort of getting biological insight. So kind of just wrapping this part of the talk up. So I told you a little bit about this cartoon model of the neuron, and which led to some machine learning type approaches. And machine learning approaches in turn can be used in biological applications to predict what makes an interface an interface, what makes a protein-protein interface. Now, this is completely independent of what the interacting partner is. So if you also know what the interacting partner is, you can further improve the accuracy of this interfaces. And this is not just an exercise in playing with computers. It actually yields some useful biological insights. So this is sort of the algorithmic thinking giving results that are actually useful in biology. So far we've talked about individual proteins and predicting maybe interfaces. Now, in the 10 minutes or so that is left, right? So let me take this one step further, because after all, understanding individual proteins or individual RNA molecules is only a first step, right? Because in a living system there are thousands of proteins, and they interact with so each protein has many interacting partners, and the function of a protein really depends on all the partners that it interacts with. And in recent years, there are experimental techniques that allow you to measure, for example, in a cell or a tissue, all the genes, the level to which they are expressed. So this can be done using microarray techniques. You can also measure the amount of proteins in a cell. So you can take this high throughput techniques, and you can actually get measurements about thousands of these. And you can also see what happens if you perturb the system. So for example, you can take a certain cell and throw different ligands at it, different drug molecules at it, and you can see what happens to gene expression as a result. So you have this massive amounts of data, and you can start building models from this. So you can so now instead of looking at individual genes or individual proteins, you can start building models of how they interact at the level of transcription, at the level of protein-protein interaction, and so on. And there are many different types of data. And so, for example, the histohybrid type experiment gives you some information about which proteins interact with which other proteins. This is noisy data, but nevertheless, it's quite useful. You can use microarrays, which, on the other hand, don't tell you specifically about what interacts with what, but it tells you something about the activity of the various genes that are involved. Okay. And then you can do more complicated things where you can measure kinetic constants and so on. So at this point in time, the larger the number of entities you look at, the coarser the data, less accurate, but nevertheless it looks at the whole system. If you look at on a smaller scale, you get very detailed information, kinetic constants, and you can actually build differential equation models, and there's a whole spectrum in between. So here are some examples of network models. Right? So this is the story. So there's some genes that code for certain proteins, and the proteins form a complex that in turn activates some other gene, and some other protein inhibits or downregulates some genes. And so this is a story, and this is a cartoon. And now you can take this and try to generate different types of models. Okay, so for example, one can come up with an undirected graph model where the nodes are genes and proteins, and the links represent the fact that they interact. And if there is also weight on the edges, then it might indicate the strength of interaction. Right? And you can generate networks like this by getting this type of data. So if you did a large-scale proteomics study, what you would get is a graph that looks like this, where the nodes are proteins and the links are the fact that they interact. So what can you do with this? Well, it's a graph, right? So if I have several different species from which I could generate these graphs, then for one thing I can essentially do a graph comparison to figure out what makes them different at the level of protein-protein interaction. So if you if you had gene expression networks, and if the links represent correlations between expression levels of these genes, you can have a graph from a healthy tissue, a graph from a tissue that has cancer, and by comparing these graphs, you can figure out what's changed at a global level. And that can then be used to generate specific hypotheses, specific, more focused experiments, try to figure out mechanistically what's going on. So, and this leads to a bunch of different computational problems. So, for example, one can take a pair of networks and align them. So, essentially, this is a graph alignment problem. And it's from a purely computational perspective, if you have no other constraints, it's also known to be a hard problem. Right? So, but in practice, in biology, there are several additional constraints that you can use to simplify the problem, and you can actually solve it. Now, here is another type of model. So here, instead of just having undirected links, you also have directed links. So you know that not only do things interact, but you know what influences what. So that gives you these types of models, and you can also have annotations of labels on the edges. For example, if this activates something, then you can say plus, so A activates itself, or minus would denote that there's an inhibitory type of interaction. And this gives this gives you this type of models. And then if you again, there are computational questions that arise. So, for example, you can ask the question about whether some gene could possibly influence some other gene in a network. Well, what is that problem? It's basically figuring out whether there's a path in this graph from one gene to the other. If there is a path, then there's a way for one to influence the other. If there's no path, there's no way for one to influence the other. So there are computational questions that computational ways of answering questions that have implications in biology. You can, if there's more than one path, then you can figure out that this gene can be regulated in more than one way. So there's redundancy built into the system. Cycles indicate feedback loops and so on. And then if you do comparative analysis, again, you can figure out what's common, what's different across different conditions. Okay, so, and you can get more detailed models where instead of just saying that there's an influence, you can also try to write down how some combination of genes influences some other to some Boolean function or something like that. And then you can test these models by manipulating directed experiments where, let's say, one of those genes is held fixed at a certain level of expression, and see what happens to that. So what we have here is starting with the undirected models, which is the simplest ones, to directed models, which have a little bit more information, to Boolean networks, which have a little bit more information. So you have this hierarchy of increasingly sophisticated models. Okay, but anytime you do modeling, you have to keep this in mind. So if you start building more and more complex models, at some point, the model becomes as complex as the thing that you're modeling, and at that point, it loses its explanatory value. The whole point of doing models is that you want the model to be simpler than the system that's being modeled, so that you can actually understand it better than the thing that's being modeled. So here's this Lewis Carroll map where the references to a map that is very accurate, and if it's as accurate as the world for which this map is being constructed, well, I could just look at the world. I don't need the map. So the map has to be simpler than the thing that it's a map of in order for it to be useful. And that holds true for the models as well. Okay, so abstraction is important, and computer science, well, there's basically a few key ideas in computer science, and abstraction is one of them. Right? Okay, so I think I'm going to skip the rest of this, which is essentially details. So let me just sort of wrap up since we are out of time. So post-Turing biology is really biology meets computer science. So computer science is the science of information processing. So computer science has very little to do with computers. The computers as we use them, that's just incidental. The key is information processing. And as long as it is information processing, whether it takes place in your brains, or in cells, or in societies, computer science has a role to play, because it's the science of information processing. And to the extent that biology has a component that is about information processing, computer science has a role to play. So computation provides a means for describing information processing. And the Church-Turing thesis, which I referred to, says that anything that's describable can be described by a computer program. This means that theories and models take the form of computer programs, and computational biology is an example of that. So we use computational models as a way of transforming biology from a descriptive science into a predictive science. So that's sort of the main idea of the talk. Missed all the details. This is one thing to remember from this talk. And this is a theme that's sort of repeating itself in other sciences. So this is not the only, biology is not the only field where this is happening. For example, if you looked at New York Times a couple of days ago, there was an article about digital humanities and how essentially using computational ideas is allowing people, humanities scholars, to discover things that they didn't know. It's historians and so on. There's also, with the advent of Facebook and so on, you now have social network information. And so you can try to really understand how social interactions evolve using this sort of data. So there is, so thinking about questions in sciences, whether they're biology or social science, using sort of this lens of computing is a fruitful direction to pursue. That's all I have to say. I'd be happy to take any questions.
I guess I'll get things started. There's been a tremendous increase, of course, in the size and computational power of computers. You intimated that it's maybe not the power of the computer, but yet the algorithms, the models. What role has the increased capability and speed of the supercomputer, we call it today, in fact, impacting what you can practically do in the size of systems you can study?
Right. So I sort of stayed away from supercomputers or the high-performance computing. It's certainly useful and important, but what I wanted to emphasize here is thinking about problems using ideas from computing. Now, there's a complementary side to that. As the problems become bigger, bigger computers help. You can scale up to larger problems, and it allows you to solve bigger problems. So, for example, some problem like protein folding, if you had a supercomputer, it would be helpful because it's a computationally hard problem. but not everything is going to be solved just by having bigger computers. The harder task is figuring out what the right modeling abstraction is, what the algorithms are. So if you know what to do, and if it's just a matter of doing it faster, or solving a problem that's slightly bigger, then yes, bigger computers are.
Thank you. Oh, is it certain that we will not have any analytical description of biological processes, so we have to, as a last resort, use computational procedures?
Well, I don't think those are mutually exclusive. So, there are cases where you have closed form solutions. Right? That's what you mean by analytical. Yeah. But there are also many phenomena for which there are no closed form solutions, and the phenomena are highly nonlinear in nature. So, in those cases, the models can be arbitrarily, so computer programs can describe the right, whatever it is that you're trying to model. Doesn't mean that you cannot understand it. You can understand the program, what characteristics it has. This is a different way of looking at it. So the computer programs are more versatile than, say, the differential equations in some respects. Model.
Technology, if I was to give you a binary protein, could you tell me what the secondary and tertiary and quaternary folding of that protein is? Just have the binary sequence of the protein. I mean, they give it the pH, and the temperature, and that sequence, and you determine exactly how it's folded.
I don't work on protein folding. There's progress being made, but it's not so. So this sort of the problem, the way I posed it, but given the sequence, can you predict what the field structure is? That's still an open problem. Okay. But progress is being made, so you can certainly you can predict the secondary structure now at the levels of 95 to 98% accuracy. Tertiary, and that used to be 60% when the field first started working on it, not that long ago. So there have been improvements in the last.
So we've got the secondary structure.
Yeah, secondary structure we can do pretty well. Tertiary structure is harder, and often so you don't do that completely ab initio starting with the sequence. You current techniques actually make use of known structures and do some comparative modeling in combination with sort of ab initio approaches. So it's a mix of techniques at this point, but progress is being made. Does that answer your question?
Yeah. I have another question too. Are there any drug companies actually creating designer drugs based on predictive models now?
I think these predictions are so one of the big challenges in drug design is you generate a whole bunch of potential candidates, and then you have to sort of prioritize them because you cannot actually build them and test them. Right? So from that standpoint, for prioritizing potential drug targets, computational methods are already being used very heavily. There may even be at this point a few drugs that are, you can trace them to this sort of an approach. I mean, not necessarily what I was presenting, but this kind of approach is that. So where you use them as essentially for screening. So screening potential targets. There's a group at UCLA, Peter Baldi's group, that has done a lot of work in that area.